these are the slides, and I would just suggest that everyone who is chairing the sub-sessions very briefly introduces their own sub-session. So Mike, would you want to start with the first one?
Sure. So this subsection's on multiomics data analysis and integration. I think that theme's come up a couple of times through the meeting. And we can think about best ways for, ooh, we lost something funny here. Anyway, the idea is some of the basics about normalizing data missing values, and then so we can get the data in a format that you can integrate. And then we would discuss what kinds of data to integrate, and from similar data from different platforms, how to integrate that. Some of the issues about single cell and bulk data, I don't know if I've seen so much of that in the sections I've been in. And then there are many different levels at which to integrate data. And I think that would make a very interesting topic. And lastly, longitudinal data, how to deal with that for integrating across time. So those are some of the topics we thought we'd handle in this subsection. Now I think we're off to Shannon's listed next.
Yep, thanks, Mike. For those of you that don't know me, I'm Shannon Hughes. I'm a program director at NCI. I help to run the HTAM program. I'm kind of filling in here as an extra moderator, so that there's anyone that's specifically interested in this network modeling topic that we've outlined. I'm more than happy to hand over the moderator responsibilities within the room. Christoph, can you go to the next slide?
Oliver might have it. Is that right?
There we go.
There we go. Thanks. Okay, so thinking about network modeling, we were discussing all the different types of biological networks and how they might be integrated to give us more information than if we studied each one alone. There's some topics on using statistical models versus mechanistic models here, maybe thinking about how to integrate into agent-based models or something that would describe a tissue or a disease tissue like a tumor. And then finally, thinking about how these networks change over time and what types of mathematics and other types of modeling can be used to describe those. So this is something that I haven't seen a lot of yet. So hopefully there are people who are interested in taking up this topic. Thanks.
Yeah, so the third topic we had, and there's a longer version on the current pages. So sorry, apologies, Christoph, for putting my extended text up late. So, but there are two major areas we would like to explore here, and that is how to deal with single cell readouts in the context of genetic perturbations. And there are two types of perturbations one could consider. One of them are natural genetic variation, so population variation, where we utilize variability on the genetic level between individuals, and there's more and more abilities to perform such EQTL analysis on the single cell level with increased cell type and cell state resolution. And the integration questions that we thought about were how to integrate these single cell readouts with bulk, conventional EQTL measurements, how to deal with discrete versus continuous cell type changes and variation in genetic regulation, but also how to sort of look at indirect effect in trans and map variants that have effect on pathways and cell states and all the way to phenotype. And the second dimension is how to utilize synthetic perturbation assays that also sort of overlaps to some extent with the fourth topic, but I think it's a very interesting question, essentially how we leverage engineered variants, using high throughput CRISPR screens and other elements, and how we maybe even can integrate EQTL maps that leverage natural variation with these perturbation screens to get more insights into causal predictions, pathways that are dysregulated, and other functional assays. So I think that's roughly everything that's related to genome and genetic variation is what this subsession will be about.
And then the final of the four subsessions will be very much focused on trying to push beyond the descriptive and snapshot character that is inherent in many human cell atlas projects, and try to find, on the one hand, integrate the time series aspect to it as much as possible, both through pseudotemporal ordering, but also through actual time series data and modeling. And secondly, to focus on the plasticity of cells, trying to look into the cell's past through lineage tracing, through epigenetic developmental potential, into the cell's current state, its plasticity, its ability to react to challenges, including like receptor ligand predictions, these type of aspects, and then the future potential. How can a cell react when it is challenged with certain external or internal stimuli? This somehow interfaces with the multiomics discussions that we had yesterday, where the question really is, how much can the epigenome profiling really add here? Which other dimensions should we integrate? But it tries to focus slightly more on the integrative analysis, not so much on the quality control and initial data processing aspects. And then the ultimate goal really is to use time and this developmental perspective in order to come to the point where causal modeling gets possible. Can we infer developmental trajectories? Can we build causal, perhaps even eventually mechanistic models of what is going on in a particular cell type, in a particular organ? And can we validate that in intelligent ways through perturbation screens? On the perturbation screening side, the focus will be on the bio, like for the whole session, the focus will be on the bioinformatic and the modeling side. But we are open throughout the 4 subtopics also to discuss experimental questions where they are really necessary and helpful in order to discuss and answer these topics. So that concludes this short introduction. Before we split into the individual subtopics, is there any, we have the option to add a 5th or in the potential case, even a 6th topic here. So if someone really thinks that under this topic, a 5th topic should be discussed, now would be the chance to pitch that idea. Otherwise, we will go ahead and start now in the individual subtopics with people introducing themselves and getting to work on those topics. Good, then I think here we are. For the next 3/4 of an hour, the goal is really to set the questions that we want to answer in the second half. Obviously, setting the question is often easier when you also give an example or already share an experience that you have had from your particular consortium. So don't feel restricted to just asking questions, but very much feel encouraged also to share experiences. I think we're very reasonably sized groups, so we should be able to really get into concrete discussions already in the first part. So I would suggest that we quickly start with one round of introduction where everyone says their name, their institution, and their consortium. And then when we are through with that, perhaps we can perhaps already integrate this because we're not so many people, already have everyone share like a single statement of what they think we should be discussing, or a fact, a technology that seems particularly relevant to them. Ed, do you want to kick it off?
Sure. Hi, I'm Ed Lean. I'm an investigator at the Allen Institute for Brain Science. I'm also affiliated with the Human Cell Atlas and the Brain Initiative Cell Census Network.
Any topic you want to share or discuss in this particular session?
I guess I'm interested in the aspect of dynamics to understand sort of aging processes and disease perturbations as deviations from that normal process.
Who's next? Sif?
Sure, Ziv Bar Joseph from Carnegie Mellon. I'm in this meeting affiliated with HubMap, but I also work with LangMap, and we've done some other work on time series analysis. I'm personally involved in some studies related to development, so that's something I would be interested in. One thing I would love to hear about, although, I mean, I have some ideas, but probably a bit future looking, but I think we'll get to that, is spatial temporal analysis. So there's a lot of spatial transcriptomics data coming in, and very little time series, but I expect that will grow. And I always say, just like nobody's talking about microarrays anymore, and very few people are talking about bulk RNA-seq nowadays, everybody's single cell. I think in 2 years, we'll just be talking about spatial. So something to get ready to.
Peter?
I'm Peter Kurchenko from Harvard Medical School. So I'm actually at HCA, HubMap, and the BICCM, but barely involved. So I think we're actually, I'm with Ed on this one. I think within this topic, I'm very much interested in perturbations associated with disease, particularly those having to do with the immune system. This is something we've run into again and again, and they're very hard to model because we don't necessarily have sort of the let's say we only see sort of the end states, you know, of the entire system. We don't see transitions as we would like, and it seems like Atlas efforts could help with this. So that's why I'm interested in it.
Okay, great. Benedict? Benedict, we cannot hear you. Can you unmute yourself?
Yeah. Good morning. So I'm from NIH. In North Carolina, so I'm an investigator, and I'm really interested in dynamics and perturbations, and with particular focus with toxicity as well, which I think, of course, most people are not really talked about, but I think it's an important area as well.
Good. Alexander?
Hi, everyone. I'm Alexander Misharin. I'm from Northwestern University, Chicago. I'm obviously part of HCA, but I'm also part of Chan Zuckerberg Lung Seed Network. My main interest is in lung biology and specifically in lung immunology. So, hi, Peter. We generate data sets both in normal lungs, so for the Atlassian efforts, but we are obviously very much interested in perturbations, both in perturbations related to disease, such as pneumonia. I think it's a great example that really unifies the response of both stromal elements in the lung, the things that are more or less static unless you really perturb the system, and also immune system that responds very, rapidly and robustly and undergoes through dramatic transformations over the course of these perturbations. But also we are interested not just in the disease, but also in the daily exposures such as airborne particulate matter or high ozone level exposures, or even smoking, right? Something that people still actually experience every day. And we are aware of some of the efforts where people actually use this to profile response of the lung at the single cell level in the context of these everyday environmental exposures. And I think another good component of the time series analysis is obviously normal aging itself, which we often do not take into account. We focus on rapidly moving developmental aspect, but we don't take into account normal aging. And I think actually how we design those studies and how do we take into account all other, factors, whether this person had flu, whether this person had common cold this year, whether this person moved into another region of the country where there is a different level of environmental exposures. I think we need to start thinking about this and how we can also leverage information coming from the public healthcare systems, such as systems implemented in Sweden or Netherlands or Denmark, where there is a default opt-in. How can we actually pull in this data and integrate it with single cell data to answer questions about exposures and aging? Sorry for the long introduction, but these are things that I'm really passionate about, and I would like to steer direction in this way.
Great. My name is Christoph Bock. I'm at the Center for Molecular Medicine of the Academy of Sciences in Vienna. So very much coming from a biomedical translational environment with very short links and direct links to the hospital. I'm coordinating one of the European contributions to the Human Cell Atlas, EU-funded contributions, which is the Organoid Cell Atlas initiative on trying to establish organoids as perturbable models where you can do all of those things that you cannot really do in vivo. And the two main questions there really are, how can we align the organoids as much as possible to what's happening in the primary samples such that we can kind of really jump back and forth between the perturbable system in the organoids and the kind of more physiological system in the human. And then secondly, how can we do perturbations in the organoids in sufficient high throughput to really make it interesting. So the topics that I'm interested in are drug treatment time series, which we've done in leukemia, and which is really interesting when you take repeated samples from the same patients as they are starting a new therapy, and then try to model really what that therapy is doing to the people. And then secondly, obviously, CRISPR, single cell sequencing, crop-seq, perturb-seq, these type of approaches. So that's all for now. Is there anyone from the people who are currently observers who would like to introduce themselves and jump in and be upgraded from observer to participant? Now is your chance. That is not the case. Anyway, you can also do that later. Then we have to volunteer 2 scribes. Who volunteers to be a scribe?
I will.
Great. Alexander?
Right. Yeah, but I need a partner in crime. You know, I'm
Yes.
terrible at the articles. English is not my first language, so you guys will.
Good. Then you have already restricted it now to those who have English as their first language, which I can probably see only 1 or 2 people. Who volunteers?
Yeah, it's not my native language either. But I can help, it's okay.
Okay, great. There you go.
Poetry.
So you write directly on the doc?
Yes.
Yes, so you have the doc, and you're also welcome to, if this helps with the discussion yesterday in one of the sessions, I felt it quite productive if if one of the scribes actually screen shares the typing.
Sure. I'll do it.
Good, then just a quick summary of where we stand so far. So I guess we should briefly go at some point down these points that we prepared in preparation for what's going to happen. In addition, what really came up strong from several of you was the aging component, which I hadn't so much on my radar, but we should definitely discuss this. And I found the spatio-temporal aspect very interesting. I think there's a lot of open questions to that, especially how to do this in a non-destructive way, and if you do it in a destructive way, how you can really connect spatial information over time if every time you sample it, you actually destroy the whole thing. So this would be 2 things that I would very much think we should be adding, but there will for sure be others.
All right, can you see my screen?
Yes.
Shall we perhaps start with the aging, because that really got everyone's enthusiasm, it seemed like.
Would you mind if I just introduce one other topic before we start? Because I actually think there are many different axes to explore here that have radically different timescales. And another one that we haven't discussed yet is the evolutionary timescale. So we've actually started now to do a fair amount of cross-species analysis. And it's quite interesting because you really see sort of gradual change as a function of evolutionary distance. And so that's sort of an extreme version of transcriptional or epigenetic dynamics, but also one that probably the same tools could apply to. Now we can go to aging if you like.
So which of the projects that we have represented here is actually doing some serious work on aging, and what are the concepts? Do you just take a couple, because you just take a couple of elderly people and you descriptively profile them, and then you hope that you see some big differences that kind of stand out beyond the inter-individual variability, or is there are there attempts to follow individual people over time, or to use epidemiological studies and go back in time, or what are the approaches that people have been following here?
I can comment on that. I'm going to apologize. I'm going to stop sharing my screen because my computer is not going to handle this. If I want to start on aging, I think we have to always take into account normal variability, right? So everyone thank you, Peter. Everyone is aware that if we do just normal profiling, even within the healthy individuals, we will often run into this problem that there is so much inter-individual variability related to sex and related to environmental exposures, related to how we harvested the tissue, that it is actually challenging to untangle the actual aging component. We have recently made a very small effort actually to tap into existing like published data sets in the lung, just to specifically ask this question, whether we can actually detect signatures related to the normal healthy aging. And not surprisingly, we were not able to find those strict signatures. So maybe one aspect actually of modeling aging, modeling normal human aging, is actually designing the specific cohorts, because not everyone ages at the same rate. You may have a healthy 60 or 70-year-old individual who does weightlifting and mountain climbing, and you can have fairly not healthy, chronologically young individuals. So when we design those cohorts, we probably need to take biological age into account. But also, maybe we should also consider taking perturbation and resilience to perturbation into account, because this is how we probably define health overall and youthful phenotype overall. We know that children, when they undergo through severe perturbations, for severe diseases, they have dramatic capacities for recuperation and recovery. We also know that young individuals also recover fairly well, but we know that for many older individuals, it's not that they, it's a smooth, continuous slope of decline. It's, what we jokingly call the cliff, right? Where you feel well, everything is fine. And then suddenly happens one event that actually changes this. So when we consider time scale analysis, it may not actually take 10 years to complete it. If we design those cohorts wisely, and if we sample those cohorts wisely before to create, maybe a repository of the samples, and then actually once we come back in a year, let's say after seasonal respiratory events such as flu, it's a big impacting factor. In any community you go, you will be able to find those individuals, and this is considered still part of the normal. If we then will be able to actually select parts of the individuals that experience this perturbation and that manage to recover from this perturbation, and then we can again sample them and ask this question: so to what extent the aging actually affected the ability to bounce back and to be resilient to this perturbation? So this is how we think about aging. It's not just slow, steady decline, but it rather ability to compensate for the perturbation.
Alex, I have a question. When you say perturbation, can we be more clear? Because there are different types with respect to this problem.
So by perturbation, we say, we're obviously not going to expose people to some, dangerous perturbations on their own. And I don't think it's necessary, because we always have these natural experiments. I think, upper respiratory tract infection that is really benign and often neglected by young individuals can be really detrimental and sometimes even fatal for elderly individuals. So I think, the disease is actually an important component, and it can serve as a perturbation. And I think it can be incorporated in the, as a way to interpret normal healthy atlases.
Okay. Right.
Did that answer your question?
Yeah, but I was also thinking about like things in the environment and things like that. So, which is fine. That's fine.
I think in the context of environment, I think, you from North Carolina, I believe NIH has a pretty large environmental studies center there. Mm-hmm. So, and I think we are actually living through this natural experiment in terms of environmental changes. You can witness how with the coronavirus related shutdown, the levels of air pollution all across the cities around the world actually dropped down quite significantly. And if we just look at the organs that are directly affected and directly exposed, I bet we will be able actually to see this difference in the natural experience.
Okay.
Hi, this is Jim Haygood, University of North Carolina at Chapel Hill, and I'm part of the Hub Map, it's part of the original lung map, and also in another consortium that is addressing this issue, which is a normal aging lung atlas, which is funded by the National Heart, Lung, and Blood Institute. And I believe there's some other folks from that consortium involved in the broader meeting. But I just wanted to say a couple of things. One is that there are data sets out there collected for different reasons at different ages, and sort of finding those and accessing those as a great way to look at both development and aging. And I think the perfect example was the opening plenary session on COVID-19, where Sasha, I don't know if you were part of that group, but a large international group got together and accessed samples across different ages to look at the receptors for SARS-CoV-2. So I think there are emerging data sets out there that are considering age and some that are specifically set up to look at that. The other thing I wanted to mention is, we've talked about biological versus chronological aging and the importance of environmental exposures. I think another thing we need to consider is cellular aging, and there can be remarkable genetic predisposition to advanced aging through things that regulate telomere integrity and DNA repair and those sorts of things. And we see that in idiopathic pulmonary fibrosis, where even in sporadic cases that aren't thought to be genetically determined, we're finding a large percentage of individuals who have genetic variants related to telomere maintenance and DNA repair. So to the extent that we can look at these things within the nucleic acid and other molecular signatures that we're analyzing, I think that may be a better place to look, or perhaps important complement to the metadata, because we won't always capture all the metadata we want to for each sample, but there are molecular signatures of environmental exposures, or molecular signatures of aging, molecular signatures of smoking, et cetera. So if we can maybe think about what are some of the key things that are already existing in the data that we're looking at that we can leverage, I think that would be fantastic. I'll shut up.
So obviously, this is the first half of this, and we have 15 minutes left. So as much as possible, we should break things down to concrete challenges or concrete questions that we can then address in the second half. So in a way that you have just done, which I think was like what I take from what you were saying is that we should have built perhaps a repository of signatures that of environmental exposures, of aging of various types, that could be overlaid with a lot of different data sets in much the same way as people have been overlaying cell cycle signatures, for example, in order to then kind of overlay an aging dimension or an epidemiological dimension on a lot of studies that don't have it integrated. And then the second aspect, which is I think as important, is the try and push everyone to collect as much metadata on exposures and aging related aspects as possible. Do you have any third aspect that we should come back to in the second session for the aging component? That would be a concrete question or a concrete task or something that where the consortia could work together to make a difference that would be beyond the reach of an individual consortium.
What I think.
Go ahead.
Christoph, good to see you. What I think you kind of put it to the point, right? You're thinking about like how to model a perturbation. This is the modeling session, right? And I think it has to be beyond just signatures. Signatures would be something that are context independent. Obviously, we do know that if you apply perturbations such as obviously aging, but even just simpler things such as a drug, it would work very differently in different types of tissue. So it has to be context specific. And I think this context specific modeling goes back to what we do, right, on the computation side with the manifold learning idea. So I think combining this sort of effect of what a perturbation would do on the sort of standard manifold of where those single cell RNA-seq data lies that we use usually to describe trajectories and so on, I think could be the underlying technique how to actually implement it. And then, I mean, in the questions later, you also, or someone put that you also want to now add causal analysis and so on. So I think this idea of having those signatures that are context specific and would sort of point in different directions in expression space, but then the manifold sort of come together, I think would be the way how to formulate this in a computational setting.
Sounds good. Shall we then move on to the topic of evolution and try to break that down to kind of concrete ideas or concrete topics that there, if there's interest to work on? I guess the main topic here, I don't know if a different consortium is a different working group is covering this, is the topic of really how to analyze or align cell types across species, like between human and mouse immune systems. Perhaps it's still possible to learn something like a shared cell space, whereas kind of going beyond that. Ed, you mentioned that you're doing this in the context of brain development. Kind of how do you cope there? Do you try to align cell types, or do you analyze everything completely separately and then do qualitative data integration? Is this a topic where you think that the people who are looking at the immune system could learn from the people who are looking at the lung or at the brain and vice versa? Or is this a topic that is relatively specialized, that's difficult to break down yet to concrete overarching questions?
Well, so I've been doing this in the adult brain so far, not in the developing brain. And there, it seems that the cell types are well enough conserved that you can do pretty good alignment across, even from mouse to human. You can do much better from monkey to human. We sort of started this mostly as a way to try to understand how well mouse data was actually meaningful for understanding human. Sorry, I have kind of an echo. Do you guys hear an echo when I'm speaking, or is it just me?
You sound perfect, but I just mute myself and maybe it gets better.
Okay. Actually, that did get better. So that actually works. It works remarkably well to align the sort of well terminally differentiated cell types across species. You know, one of the really interesting things is that we've kind of had a chance to look at this in more detail, is that if you look at sort of cell type specific signatures, the majority of genes that have really selective expression are actually changing their expression patterns. And yet the minority are really well conserved, and you can use those to align. And that's kind of, I think, open up the possibility of quickly honing in on these really key, well-conserved types of genes and transcription factors and regulatory networks that must be responsible for why there's sort of this superficial level of conservation. But at the same time, we can understand now, especially as you bring in more species, how these things start to diverge from one another.
So Ed, are we talking about static alignment or dynamic alignment? Because do you have dynamic data, or is it just
static data? Yeah. So dynamics would be even. I mean, that would be great if you could do an evo-devo and bring in the developmental axis. We just don't have the data for that yet, but you could see that coming.
So Ed, how is this different from bulk analysis? Because a lot of people did already a lot of bulk evolution studies. I mean,
Yeah,
shouldn't we focus more not just on the genes, which we can see in bulk, but on the cell type, for example, or they conserve the proportion of cells and this kind of thing?
Oh yeah, so all of these things change, and you can study all of them with the single cell data. So you have sort of this general conservation, but then you have species specializations of different kinds of cells. You have differences in proportions of different kinds of cells. You know, it's, I mean,
sort of a better conservation when you look at the single cell level versus the bulk, or is it the same, but you see other dimensions? Like, can you contrast these? So, I mean, at least that's something that would be interesting.
Yeah. You know, overall, I would say I'm struck by the level of conservation that you see at the cell level, which really argues that at least in the brain, that this architecture is extremely well conserved and under evolutionary constraint. But then, you know, when you get into the details, there are lots and lots of differences all over the place. So I mean, it's basically like every other phenotype that you can study. It's just this is a very rich one that's you know, where you're kind of getting at the root genes, the causal genes for changes in phenotypes. We'll have two papers going out onto bioRxiv in the next day if you guys are interested in kind of learning a little bit more about it. But you know, it's quite complicated because now instead of bulk going in one tissue, you've got 100 types of cells that you're trying to look at simultaneously, and some are better conserved than others. But it's extremely informative kinds of data. But part of why I brought the topic up here is that you know, of course, right now most of the data would be kind of your model organism versus human, but you can also use this to kind of study evolutionary processes if you bring in more species, and that's something that we're just starting to do now. We're trying to bring in about 20 different species and look in one part of the brain and see really how things change as a function of lineage.
Good. Before we go into the break in like 4 minutes, let's briefly discuss about time series analysis. And obviously with the 2 dimensions of pseudotemporal ordering that a lot of people have done on static data, single snapshot data sets, but also with the actual single cell time series that people have done in the hematopoietic system, but potentially is also getting traction in other fields where it's possible to actually do repeated sampling. That is a topic that has received a lot of attention for computational modeling and manifold learning, and kind of how much really, like, has anyone kind of systematically looked at how much the time dimension, if you actually look at a repeated sampling, adds on top of like a pseudotemporal ordering dimensions, or are there any analytical questions that are not yet addressed through kind of extensive work in this field?
I have another. Does anyone know if there are plans to develop dynamic data sets at the single cell level? Because I still feel that it's still very difficult to get this kind of data sets. I've seen people always talk about it, but I still never get to see the data sets. And it's always, we talk about it, but people don't generate it still.
I mean, there's quite a few in literature, right? And there have been a few methods, such as the ones on optimal transport, or we had one we called pseudodynamics that have been working on that, typically for development or for maybe reprogramming or something like that.
But they're not stochastic. They don't, I think about, yeah, maybe it's because it's, but they don't really control for stochasticity. That's the problem as well.
What do you mean control for stochasticity experimentally, or what?
I mean the transitions. Yeah, it gives you a sense of yes, the model, the transport of cells, but I think there's still like, it's difficult to quantify uncertainty and kind of like in terms of, that's the problem with those methods. But they're fine. I think they're good, but I just feel that sometimes it's really like validating them and things like that.
I mean, in any case, I think Christoph put it right? There's like two angles to it. Is like with the cross species, in a sense that if you have multiple cross species, you could ask different questions than just mapping everything to one particular model organisms, like Ed said, right? You map everything to human. Well, then it's easy because then you just have those pair type of things. But as soon as you want to have 10 time points, maybe even in your case, Ed, I guess sort of different type of species potentially being sort of different axes on the tree. This type of integration is harder, and I don't think there's so much has been done. So everything I know from the cross species for sure is just two on the single cell RNA-seq, and for the time series, it has been mostly people just throw everything together and then later just see where the time points lie. Right?
Yeah, but in a way that is, I think, because we haven't invested enough time yet into procedures that can model the heterogeneity of cells while also adding the modeling the heterogeneity of time as separate dimensions. So kind of what people then do is they just simplify everything by essentially projecting everything into a single UMAP, TSNE, whatever space, and then show videos how clusters go up and down as cells are responding to a stimulus. And part of the reason I think Benedict is right in that sense that we don't have a lot of good single cell data sets that have a real time series component to it and have more than 2 time points. There's a lot of kind of pre-post treatment, but really something where on a relatively dense time scale, you have like 5, 6, 7 time points. That has been done mainly for relatively artificial in vitro systems and not really done a lot for kind of human samples. And there, I think quite some value lies in analyzing clinical trials where patients get a relatively strong drugs given at a given time point and then go through a trajectory of effects and side effects which could in principle be sampled.
Well, I think you're right that there's kind of a dearth of data sets with dense time point sampling. And there are not many methods that are targeting this. I think the one obvious exception is the work from Lander's group, right, about the optimal transport thing, where they had fairly dense time series to go with this. But I actually want to bring up a slightly related but different topic that will relate to both time series and maybe aging, because I think the challenge is that we're not going to get these perfect time series, right? Densely sampled ones, not often anyway. So the question is, can we have methods that are able to assemble this time series from basically data that has inter-individual variability? You're just sampling, let's say, a large population of people. You're not sampling them maybe like once, twice each. So not very densely, but they all lie somewhere within that spectrum of a time progression, let's say aging and so forth. So can we come up with methods that are robust enough to reconstruct the time progression from this type of data? Because I imagine we'll see that more often than a nicely designed, densely sampled one.
Peter, what are the specific challenges, from your point of view, working with these data sets? Is it just experimental design, let's say, like how we collect samples, how we ensure the quality of samples, or there are specific computational challenges that you envision here?
Well, I'm sure they're both, right? Obviously, any technical variation that's added on top of it makes things more difficult, especially if you don't have the metadata to control for it. However, even if you measure it in a perfect way, still inter-individual variability is major, right? So if you say, let's say, each individual, let's say we're talking about infection, right? Like a progression of disease. And if each individual sampled with 10 time points, you'd probably be able to look at individual and then reconstruct it, right? Well, let's reduce it. So we sample them, each individual 3 times or 2 times, or maybe even once. So then basically disambiguating inter-individual variability from temporal progression and where they are in the pseudotime becomes more and more challenging. And I think it's just, I mean, we can solve it, right? But we don't have methods like this.
I mean, we recently published a paper on plasticity in EMT yes, it's really difficult. But those kind of data sets, like the ones we develop, we used, they can really bring into light some of the properties in dynamics that are not very obvious, like processes that don't go back like in the same way, things like that. So sometimes we might have to do the fine grain to kind of capture some of those states and cell types, which will not be captured in the traditional methods.
Good. How shall we organize ourselves? Shall we go into 15-minute break now and then switch to problem-solving mode after the 15-minute break, or do we need to stay with the questions for a bit longer? Any opinions?
Maybe just a few minutes longer. I just want to maybe raise that we could treat the disease progression as a time series as well. That we could have instead of pseudo time, a pseudo disease progression using these same sorts of things, and that might actually be more robust than aging, which has so many other uncontrolled variables. Maybe that, I don't know, and I don't know if that will apply to all diseases, but like a progressive set of diseases like neurodegenerative diseases, maybe that would work pretty well in.
Right, and this also works in lungs. So we recently did some study where you see different areas in the lung with different progression, and you can treat it as a time series in the same way. And something that is almost impossible to do in real lungs. You're not going to get biopsies for people every few days. Yeah, I think that's a solution.
I agree, and I think that also looking into disease will also allow us to bring the perturbation, the control perturbation as a drug, because we know that we're targeting specific pathways. I think that would be nice way to perturb system in a causal way, or as causal as it gets.
But kind of seeing what has been said so far, one question comes to mind: is everything a time series? Can we, in a way, with the same computational methodology, model, so some form of manifold learning that is sufficiently abstract that we could, in principle, plug into it a very short-term stimulation of stem cells in the dish, all the way to the drug response in an individual, the disease response or the development of a disease in an individual, the healthy aging in an individual, as well as the evolutionary dynamics? Do we think, like, where would that break? Is there any reason to believe that some of these things are so qualitatively different from each other that there could not be an underlying computational methodology that would be reasonably suitable to model these dynamics?
In my experience, the evolution looks different. It's just the changes are very they're structurally distinct. They're systematic. I don't know. Maybe it's just an extreme case, but
their stuff base is not overlapping, right, Peter? Is that the problem?
Yeah, I mean, actually, I mean, Ed pointed out that things are surprisingly conserved, but to me, looking at this data, I was like shocked how diverse they are in terms of actual expression programs that are happening. I mean, this is like mouse and human even, not that far if you look at the sequence, right? So I can't tell for sure whether this is, obviously when you kind of, after you establish the initial connection, I guess we're talking about like pseudotime sort of things here, right? Then you dive into specific things. And in the evolutionary question, you're gonna dive into like connections with sequence mutations and so forth. So after that, I think paths diverge. But your question is, can we have this initial algorithm that will bridge, that establishes pseudotime in a common way? I think either evolution is different or it's just a very extreme case.
So if I can just change subject a little bit, what I've been thinking about doing, because we have these data sets where we sample the lung, so we work on lung, basically along the tracheal tree or the bronchial tree from proximal to distal. So there's there's a zonation, if you wish, in that. That, you know, analyzing that using some kind of pseudotime method might work as well. So their time, or your, you know, your parameter is no longer time, it's actually distance along a gradient or something. That should work as well, right? But I'm not aware that anybody has ever done it, or I might have missed it. So any input would be nice.
I guess that was the point that was brought up in the very first introductory session, whether we should really start thinking about the spatiotemporal analysis, where we would eventually have to bring in the different dimensions. And it seems that there is a developmental dimension that could be considered a temporal dimension, but also has a spatial component to it in many cases, where there is the stem cell niche and then progenitor cells going out from there. There's definitely the temporal aspect of responding to challenges, stimuli, and there's definitely the spatial aspect. So it seems that these three aspects, the kind of short-term temporal, the spatial, and the developmental, that are axes that in most tissues would need to be modeled more or less simultaneously.
I have a comment. Is this modeling going to be within the context of future predictions, or is just going to be for a particular application?
I guess for now, the focus is on understanding how things kind of belong together, like which cell corresponds to which other cell, how does the regulation change in response to stimuli. Obviously, prediction is a big step beyond that, which could then be validated with perturbations. So that is yet another dimension where you would essentially try and infer cellular plasticity and the ability of cells to contribute to an immune response, to contribute to regeneration of a tissue. That seems like a second step that's going beyond getting these first three dimensions of space, time, and development under control.
Okay.
Good. Are there any kind of tasks that come to mind, like concrete topics, questions we could work on in the second half, where it's kind of realistic that we'll make some progress, and there's also some value by integrating across the consortia?
Ziv, if you're online, maybe you can comment. I believe you are part of the NALCA consortium with Naftali Kaminsky that focuses on aging. Maybe you can, from the consortium perspective, say how we can integrate this information. But I understand that it started only recently, so you may not have a lot of,
Yeah, so I guess the only thing I would say in that regard is that your suggestion to design the cohort is a great suggestion. Unfortunately, if you're dealing with lung or kidney or other things, it's almost impossible to design the cohort because you are basically restricted to the samples you can get. And these are either post-mortem or related to transplant, which are very sick people. So I actually like, if I would focus on something, I like the idea that was, Peter and others mentioned about this, trying to come up with a very global alignment or pseudotime or whatever, where we can actually take the normal samples and somehow, or even not normal, but somehow extract the normal component and try to put everything within a joint reference. And then, for example, for aging or for anything else, because designing a good cohort for human, I mean, for mouse and other experiments, I can see that, but for human experiments, designing a good cohort, other than blood samples or maybe some other simple things, is going to be extremely hard. That's my impression. In terms of NALCA, right, I mean, we have, basically, we have a set of individuals that we are profiling, but it's almost a given set of, it's not like there's a lot of choice.
I mean, I was just saying that in the field of toxicity or toxicology, they are moving away from even mouse models to even in vitro models. And so I still see the use of might be still optimizing some of these developmental processes at even using non-human systems, and then trying to extrapolate them to humans. I guess that is one way to kind of like mitigate the limitations in both systems, because it's really hard to get the human data. But I think if we can use non-human data and try to really optimize and model them in by generating very good data, we should be able to extrapolate to to the humans using static data also.
I think this is an excellent suggestion, and several groups have mentioned using non-human primates. I believe actually in Germany, in Munich, Herbert Schiller has established some work on working on baboon lungs. I think that might be one way actually to potentially extrapolate this. It's I still think we will have to make those baboons travel and also learn them, teach them how to smoke. But going back to the issues with obtaining human tissues, I would actually, and I think Martijn can comment on this because he has done some work in this field. I think we can consider using easy to access tissues such as skin, right, which can be accessed and can be sampled relatively easily and painlessly. We often do this in different clinical trials. And also accessing the nasal epithelium as a surrogate for the respiratory tract across different ages, because those tissues can be sampled relatively easily. And recent work from Pascal Barber's group in France and Martijn and Sarah Teichmann, they also have demonstrated that transcriptomes of the individual cells, and this is actually where the single cell really comes to power, because you're no longer averaging in bulk, but you're looking at the transcriptomes of individual club cells, ciliated cell, basal cell, and you can ask those questions. Can you actually detect age-related signatures? And those samples are very easy to obtain. Nasal biopsy is actually much less painless than blood draw. So I think there is actually room in this direction, going for surrogate tissues or going for the tissues that are easy to access, and also tissues that are actively exposed to various environmental exposures.
I guess you want me to say something now, Sasha. I agree that nasal epithelial cells are relatively easy to obtain. We even don't use biopsies, we use brushes, which is even less invasive, and it works great for getting the epithelial cells. And yes, you could model, or you could actually sample this longitudinally in individuals. So that's actually what we are thinking of doing. We haven't done this yet, but it's definitely a great idea. It's just an experiment that takes forever to perform, of course, because we're working with humans. But even if you do it just cross-sectionally, taking aged versus young nasal cells, there's a huge, there's a huge amount of difference there, which you would expect. But then again, that's hard to extrapolate. So and do time series on, etc. Because the individual donor is still the major batch in that data, more so than anything else. So there's there's still room for improvement. Let's put it that way. But definitely something we should explore, I think.
Good. Coming back to this question of how similar or distinct are the various ways in which time can be modeled? Do we have sufficiently, given that there's been a lot of investment into kind of manifold learning on single cell data sets, do we have sufficiently generic, user-friendly software where people can throw in an aging data set in pretty much the same way as a short-term time series stimulation data set, and plus perhaps all kinds of other time series, and get something out that would be kind of the best of the abilities of this generic piece of software at reconstructing something like a timeline? And if so, can that be provided if we can combine such software with a lot of different data sets from very different types of time series? Can we learn something about kind of the fundamental equivalency or non-equivalency of the kind of modeling of these various types in which time series can be obtained and tackled in the single cell field?
I guess the question is what you want to achieve, right? And if you want to just put things together, I think you could learn a joint embedding, a joint manifold or whatever. But what do you want to do with these things? Do you want to potentially interpolate? Would that be one question? Do you want to maybe mechanistically understand, such as I think Ed sort of laid out, to see maybe things that are common, that are different between species or in a time series? Do you want to understand which genes or something that stay constant or so? So I think depending on the question, you might really want to maybe do different things. So I don't think it is necessary to maybe even map everything onto one thing. Maybe just differential analysis would be fine, no? So I think just answering without what we want to do afterwards might be too hard or too generic.
I guess my point is, at the moment, it seems still kind of very much like a generic type of approach to first put everything into some form of UMAP, TSNE, whatever, and then you project on top of that everything that happens in your system. And the question is, is this still widely used, relatively generic approach? Is this the best way we can do as a first go? Or could there be some form of similarly generic learning that could be informed about key variables of the sample collection, like time points, and then you would map all stuff downstream on top of kind of this learn manifold rather than on top of a simple UMAP or TSNE projection?
I mean, Eric, right? Eric had this Waddington OT paper, and we had sort of parallel on bioRxiv this thing where we had those population dynamic model where we basically see how one population, let's say maybe early stem cells, is maybe contained across different real time points, but there's maybe some additional development branch going on, so that you have a non one-to-one mapping of real time to pseudo time. And we call our approach pseudodynamics, was a Nature Biotech paper last year. So there's these few things have been around on the population level, just to go beyond, as Christoph rightfully says, just seeing how the UMAPs look, but really to see how populations flow into each other. And I think Alon Klein had something similar on where to then see sinks and sources where cells are being generated and where they are pushed out and so on. So it's more on the population level side. So I think linking this, I think, could be extended, but that's something that I think people can do. But then how to map then also on seeing what's happened on the transcript level, I think this hasn't, to my knowledge, been really done, because where usually those things just basically link this cell graph type of representations.
Good. We just got the reminder that we should slowly but steadily move to problem solving. So to kind of, so my suggestion would be now that kind of those people who are actively participating, or anyone else who wants to start actively participating, says 0 to 1 concrete topic where you think we should, in the second half of the discussion, try to bring it to a concrete goal, next step, deliverable, something that can, where we may be making an impact by bundling our energies. And then we go into like a 4-minute break, just people can get their new cup of coffee and change their mindset from problem identifying to problem solving mindset. Everyone can stay in the call, so we're not closing the call. And then we, in kind of after that, we would try and solve those, at least some of those tasks as much as possible. So shall we go in the same order as we went earlier? And Ed, you want to kick off with a concrete idea of something where we may, like an objective, an outcome, something where we may achieve traction in this group that would be worthwhile then to be followed on by some of the people here plus people outside to get to a point where we've created something that is useful for the community?
Yeah, well, I guess, I mean, in the spirit of something we might actually want to do later and that capitalizes on the talents and the different data sets, it seems like there are a lot of data sets that the different consortia could contribute to testing the generalization of time series modeling, or maybe the improvement of that at various scales, and test this idea of whether there kind of are some generic things that could be applied across many different systems. And I think there are now data sets that could be used to do that if people had the appetite to do it.
Great. Who was next?
I think Ziv was next.
Well, sure, I like the idea. I mean, if I understand correctly, so creating not just a method, but I think one practical issue that can come out, and this is something that, you know, we observed back in the days of microarrays and RNA-seq, and that time series is often mixed with other things. So the question is, can we create a repository for time series data only? And we have to define if 2 points is time series or not. But single cell time series across animals, across conditions, but really dedicated to time series, which I'm not sure there is.
Peter?
I think we can probably make good progress on the aging question, because, well, everybody has an age, and all the samples collected, you know, are sort of, can be certainly tagged this way. There are plenty of, you know, work that's been done on sort of using DNA methylation, other molecular techniques to assess the biological age, and it could be linked with some of this. So it seems like an addressable problem that could largely use the samples that are already being collected if we just get some extra experiments, extra metadata once in a while. And also it touches sort of on other things we discussed, like reconstructing those progressions from, you know, inter-individual variation data, from other things and so forth.
Alexander?
Yeah, I'm going to continue on this topic of what we started in temporal analysis, but not from the point of aging, but rather from point of different outcomes. So if the system is perturbed, and I think it's particularly important and actual for immune system, but maybe also for some epithelium-rich organs as well. If the system is perturbed, you know, like, will it actually come back to the same state, or will it form some new stable state in response to this? And we often see this both in the disease and after perturbation. I think these data sets will be very, very informative, where we actually can link the specific, you know, single cell signatures to the known clinical outcomes.
Good. Anyone else? Good. Then it seems that two topics have crystallized as a high priority. One, it would be time series with 3 components. The first one is what is a time series, and can we build intelligent repositories of time series data, which then kind of goes into the second point. Can we validate in a meaningful way those algorithms that are already out there, compare them, see where they are breaking, etc. And then the third aspect is, can we develop guidelines from that, what people should be doing when they are facing something that has a dynamic component to it. And that would go from the simple differential analysis or a simple static snapshot of a tissue towards something more dynamic. So that would be the first point, with a very strong methodological component and also some benchmarking character. And then the second topic would be aging. How can we make sure that aging is integrated in as much as possible into as much as possible of the human cell atlas, on the one hand through metadata standards, and on the other hand, through signature plus context, like that would essentially add this, add or extract this aging component as much as possible for kind of existing data sets. So that would be the 2 points that I kind of tried to crystallize from what you've been saying, but please feel free to add any further points that you have before we go into a very short break.
Hi, this is Jim. I just wanted to throw one other thing out there as we're thinking about these problems is maybe start thinking concretely about what, if there's a context that we can look at some of this in. And one thing that maybe is unifying some of what we're saying is there are several systems where epithelium is in contact with the environment. We've talked about skin, we've talked about lung, we haven't talked about gut, but that's another one, where you have a lot of environmental exposure of epithelium and then probably communication with the immune system. So it might be interesting to look at some of those barrier tissues as test cases where you have both age and environment as stimuli that are occurring naturally, and then seeing if there are generic ways to capture that work across tissues.
I think that very much reiterates something that Alexander mentioned earlier that I had kind of forgotten here, that the aging component also should have very much of an environmental exposure component. Some of us are back. More of us are back. Okay, I think that we can declare critical mass, and certainly Peter has gotten a coffee, which is what the coffee break was for. Let's ultimately, what we have to fill in at the end of the session is we have to fill in this blue template. We have now 40 minutes until the end of the session, and by the end of that session we will have to provide something reasonable on the blue template. We don't necessarily have to fill it out completely, but we should have a very clear idea what we want to write there, such that we can then in the last minutes after the call just very quickly clean up our notes and shift as many things as possible, or those things that make sense into this section. So we will have to give a title of whatever outcome we want to put forward. We have to define really which problem or problems we want to be solving and how we're going to do this. And clearly here, it's better to put forward only one or maximum two things that we really think we want to do and we're going to have an impact with, rather than a lot of things that are just spurious notes, because the notes we have in the main part. And then obviously as was discussed in the plenary up front is that there will be a call whether we want to give a 2-minute summary of what we've discussed here in the plenum, which has to go by relatively high standards in terms of we actually wanting to do this, and we having a concrete plan, and we have questions for the group as a whole. So these things we should keep in mind now as we are trying to work on these fill out more details on these topics of time series and aging that we have identified in the first part of the discussion. Does anyone feel like, for example, aging is definitely a topic that didn't originally come from me. So is anyone wants to kind of take the lead for structuring the aging component into something kind of meaningful and also sufficiently tangible that it can be taken forward? Good, since Ed hasn't really turned his video back on yet, so let's perhaps start with the time series then. Okay, there he is. Because that was certainly one thing that you were also advocating, but I don't know, shall we start with time series? Shall we start with aging? Aging seems to be a bit more concrete and a bit more focused, so I would perhaps suggest that we start with aging, where chances are a bit higher that we can reach a kind of very concrete results of this discussion. So one obvious thing that someone could be doing in kind of perhaps this year, perhaps next year, perhaps the year after, is to try and do a meta-analysis of aging in the Human Cell Atlas, where one would pull out a lot of the data that is there together with the metadata that comes with it, and try to find whether there are tissue-specific signatures of aging, whether there are kind of overarching signatures, whether the signatures are shared, underlying similar processes, etc. And one outcome here could be that we provide some form of kind of initial platform infrastructure, recommendations, standards, etc., that would make it easier for those people who will eventually go along those lines to do this. Or obviously in the most extreme case, someone could already volunteer and pitch the idea of such a meta-analysis at this point in time. Or perhaps there are even consortia or individual groups that are already trying to do a meta-analysis beyond lung cell atlas or brain cell atlas and really try to get at these points. So these seem to be kind of different levels of granularity or ambition, just preparing the grounds that other people can do it in the future versus trying to already set up some form of plan versus kind of immediately starting to do it.
I wonder if it might be helpful to think of what the sort of end goal for something like this would be. I mean, why do we want to do this in an atlas context? You know, one use of these is a reference that people can map their data to. And so if you can define this trajectory, then the goal would be that anyone else could map to that trajectory with smaller data sets insufficient to do it on their own.
I guess there are large communities interested in aging, and a lot of that has been focused on individual tissues, but there's potentially a lot of value in looking at how aging signatures are similar or different between tissues, which seems like something that would have to involve some form of meta-analysis across consortia at some point. And since most consortia, at least is my impression at this point, do not have a major commitment yet to doing a systematic aging research, if we were just taking, kind of letting another year or two pass and then taking all the data that is there, perhaps that would not give rise to a meaningful human aging atlas, or because we would just not know enough about the aging status of these various samples, unless we can somehow reverse engineer it based on signatures.
To follow up on the concrete recommendations, because I think here in the States, these recommendations would be read by people from NIH, our major funding agency, and they would look for these recommendations in terms of shaping requests for applications. So in terms of time series analysis versus normal aging, would we actually propose our ideal cohort to be real time series analysis in aging individuals, and actually we can design the collection of the tissues from these studies? Because they don't have to go into single cell RNA-seq now, but we can actually acknowledge that it's a challenging problem to address, and we need to design very careful study for this, and then we can design such study and say we will sample these tissues that can be sampled repetitively from these individuals, and we'll access, interrogate them using these and these techniques that will be still actual, or maybe there will be better, techniques that we can use in 2 or 3 years once we actually collect across substantial time series.
So then essentially what we should add under aging is a third point, kind of what would be a reasonable design for a kind of the ultimate study on organ-specific aging in a human cell atlas context.
I think that there are two questions being discussed, right? One is just a passive sort of signature of aging, if you will, right, from a homeostatic kind of view. And then the other one, which Alexander is pioneering, is a study of a response to essentially an insult or some sort of perturbation, right, as a characteristic of aging. And obviously the two have different designs. The first one is probably easier because we could, with some extra data, we could collect it passively, more or less, or piggyback on the existing designs, right? And the second one probably requires targeted design, and that will maybe a recommendation for NIH or other funding agencies.
So those people who have already done aging studies, how do you just collect chronological age of the people, or do you do any physiological assessment of the individuals? Do you kind of do an epigenetic clock, biological age analysis? Do you do anything else? Could you imagine, like if you were planning such a studies, what would you want to know in terms of metadata?
I think as an example of collaboration, I think we can really tap into existing large cohorts, particularly cardiovascular cohorts. So I'm willing to step out a little bit from the lung realm. But I think one big example that is perhaps widely known is a Framingham study. And it had its own limitations. It was a great study, but it had its own limitations. And now it's actually was extended into large cohorts, such as cardiac cohort. Cardiac cohort has been going on for 30 years, and they actually have absolutely astonishing retention of the individuals that were enrolled into the study 30 years ago. They do have some amount of biological material from those individuals. Unfortunately, it's mostly frozen blood, but these individuals are now coming for follow-up. And what we do have on those individuals is very rich clinical metadata. So we can ask specifically what happened to the biological age of these individuals, right? They're all, let's say, 60 years old, or they're all, 40 years old, but what is their biological age and how well they actually function? And I think for organs like heart and lung, this is extremely important. What is your cardiovascular health? What is your lung health? And we do have people in our group, Ravi Kalan, in collaboration with Avi Spiro's group at Boston University. They are asking specifically these questions in the cardiac cohort, which is cross-sectional cohort, but they do have all the time series analysis on the clinical parameters. So maybe if we actually propose to tap into existing cohorts, and such cohorts also exist in Europe and Asia, to ask these questions, right? Even if we are doing sampling for, let's say, our atlasing purpose only at one point, but how can we incorporate all this rich clinical metadata to inform that kind of analysis?
So essentially what you're saying is we should add now single cell resolution as a new modality into kind of when large long running cohorts are being resampled, in order to, then that will not give us a time series of aging at the single cell level, but it will give us a single cell outcome of a 30-year long process of physiological aging.
And I think this actually resonates with what Peter was saying earlier, that if we cannot design very dense sampling of the samples, and we can have maybe 1 or 2 samples across the time, or we can have known ages, that are really spread out across the whole continuum, we can actually try to infer those signatures from that kind of analysis. So that would be composite analysis. Peter, am I right? Did I get you correctly?
Yeah, I think so. I mean, I was mostly thinking about HCA collections, but I agree. If we can sample some of those individuals from the ongoing studies, that would be great.
But that brings kind of the components quite nicely together, because we would want that whoever runs a classical human cell loss project to capture aging-related metadata, which we would still have to define. At the same time, we would want aging studies, such as large-scale cohort studies, to add a single-cell readout as much as they can. And then the signatures would essentially be the one thing that connects the two things. So we would infer signatures from the human cell atlas, where these signatures would be very high resolution in terms of the cellular and molecular microenvironment, for example, whereas in the cohort studies, where the profiling would be more shallow, they would give very accurate information of past environmental exposures. And then essentially we could connect the depth of the human cell atlas with the kind of time dimension of these these cohort studies through signatures that would be mapped one way or another, and would annotate kind of one data set based on the reference of the other.
Yeah, that sounds good. But I was thinking also, how about I just a kind of a background suggestion to have a common like DNA methylation sequencing for estimation of biological aging. That would be kind of centralized, I guess, for HCA or some of these NIH efforts. So like if you're not really doing the study, but you're doing a sample, and to encourage people to send in a little bit of that sample for that type of analysis, just so that, you know, people who are actually interested in it can get that as a data point.
I guess that's quite challenging for a lot of people, because that would mean that they have to collect peripheral blood on top of whatever they're analyzing. But it seems to be good practice to collect peripheral blood anyway. And that could be a good, or could be one very specific reason why people really should be doing that.
You can do DNA methylation signature of aging in tissues as well, but that sort of begs the question of is the signature going to be the same in each tissue? Are there going to be inter-tissue differences? And should we have a reference tissue like blood as something that ties everything together?
Good. So regarding the metadata, what is the minimum? Should there be something like a minimum information on aging that so what would be included there? Biological age, cardiovascular health, you've already written down. Can we make this sufficiently concrete that this is something where we would have at least kind of a short list of doable points, and whether the epigenetic age based on peripheral blood, which is standardized, is doable or not? That would have to be left for the individual consortia. But do we feel competent here to spell out this list in sufficient detail, or is this something where we are really lacking the expertise and should leave that for a different venue?
There are many discussions of metadata within this conference, and so just making sure that we communicate with the other groups that are looking at that, and be sure that there are some important aging hallmarks that are captured.
Okay, that seems like a concrete outcome to do, which is to connect to the metadata group and see if to make sure that enough aging information is included there, and see if that would also be a way to sneak in an epigenetic age based on peripheral blood samples into some metadata standards documents of the human cell atlas, which makes it more likely that people actually adhere to that. Shall we spell out a bit more what's point one here of the recommendation? What would be the design of the ideal aging study? Or is this something that is not really a good use of our time now? The alternative is going to the time series.
I think it's worth recording Alexander's idea about just studying aging in a context of a dynamic response of some sort, even if we don't propose a specific design, but just mention that this seems like an important feature that's not obvious.
Peter, thank you. Actually, I think the design for this study is relatively simple, and actually, I'm aware of a couple efforts. That doing exactly what we were just talking about. A group in Canada, they're sampling young individuals and aged individuals before and after winter flu season. That's a simple design as it gets, because we know that approximately 20% of all individuals, both young and old individuals, they will experience any sort of, upper respiratory infections, and they will recover from this in a very different way. And they're collecting, they're doing exactly what Peter was just saying, collecting blood for DNA methylation, and they're also sampling nasal epithelium as a surrogate measure for the lung health, or just actually epithelial tissue that is different from peripheral blood. So actually, I think that studies like these are very achievable, and it's a good component with perturbation, and you can assess the function of particular organ of interest, whether you're interested in, Ed probably would be interested in cognitive performance or in some other parameters. This all can be incorporated in the model.
I think that's a very good point. So aging is not something that is just static, but something where the interest is really in the functional implications. So aging should be studied with a focus on the plasticity and the perturbation response that is inherent in it. Because ultimately, this is also why aging matters, not because people look different when they're older, but because they have reduced plasticity and reduced ability to cope with certain environmental challenges, such as the flu infection. Good, that seems that we are, for the aging component, actually kind of reasonably far. So we should, the scribes are currently writing this up, which is great. So we would have kind of one concrete comment to how future aging studies should be or actually two components. One is that they should, if possible, include a perturbation component, or second aspect, they should build on those cohort studies where there is a long information on the past, like cardia, like Framingham, etc. And then the second aspect was really the metadata, and where we would reach out to the metadata groups of the Human Cell Atlas and make sure that they have enough aging-related metadata included there, and ideally via that path also emphasize the importance of epigenetic age as a form of metadata that needs experimental assaying of peripheral blood, but that would be very valuable for kind of integration and age-related analysis within the Human Cell Atlas. So it seems that this point is quite concrete and well documented. Do you have any further additions, comments on that before we move to the time series?
Could I just ask, what is the status of the epigenetic clock at the single cell level? Is much known at this point?
I don't think there has been much done on it. The problem is that DNA methylation is still very, very inefficient to do at the single cell level. And since the clock in most of its incarnations is based on the Infinium assay, which is absolutely a bulk assay, there's no way you can do Infinium analysis on even close to single cells. I'm not aware that really anything has been published on that regard.
Has anyone compared DNA methylation versus chromatin accessibility in a very controlled way to look at whether that anything can be extrapolated from chromatin? Does anybody know about that?
I'm also not aware of such a study. Or you would assume that if you have done and run your epigenetic aging clock on 300 individuals, and now you also have a TAC-seq for those individuals, it should be relatively straightforward to build in a TAC-seq-based epigenetic clock, and that one would then be possible to project on top of the TAC-seq data that is perhaps collected more frequently at the single cell level and in much more throughput. But at this point in time, I'm not aware of someone having done that.
I think actually that the TAC-seq is much more dynamic than methylation in bulk studies. I'm not sure about single cell, but in bulk, certainly it's much more dynamic.
Can't really hear you, Ziv.
I think that the TAC-seq is much more dynamic than methylation in bulk studies. So it changes much more rapidly, more in line with expression. Whereas methylation is more static, at least, again, bulk and right, depending on the condition, but that's at least I think what's coming from bulk. Whether this is true in single cells or not is different. I don't know.
Good. Any further comments before we move on the aging part, before we move to the time series? Good, then let's continue with the time series. So what I wrote down here, and so Alexander, we just, do you have any final comments on the aging before we move on to the time series? We were just about to switch.
No, let's go to time series. I apologize about that. I had to step out. We have a clinical study. I'm like the only person covering for 5 people. Sorry.
But for the time series, we would still need to break things down much more concretely. What I wrote down here as points capturing from our discussion. Obviously, this is far too much than we can realistically achieve, but in a best of all world, we would have a very systematic way to collect everything that looks like a time series and put it into repositories, then run a lot of algorithms for time series modeling against such a repository of a lot of single cell data sets. We would compare their performance. We would learn a lot how these different algorithms perform on these different data sets, and we would from that extract guidelines and best practices that we would share with the Human Cell Atlas and the broader community on how to analyze time series data. Obviously, that's something that seems like a project for 10 people for 2 years. So perhaps that's a bit too far reaching. But if anyone wants to volunteer or take the lead for any of these specific subpoints, or even for the point as a whole, that's definitely appreciated.
I guess I would just make a comment similar to a previous one, that there are the things that can be done sort of pro bono, and there are the things to identify an unmet need where the funding agencies might want to create opportunities. And I would, if you would look at these sorts of things, it seems that some of the first ones could be done, of sort of canvas what's there and do some pilot work. But then, to your point of needing 10 people to do it, ultimately we need resources to do it, and so that could lead to some recommendations for future funding of developing these types of algorithms.
I am actually starting in, at NIH, so I'm really also kind of interested in this kind of problems. And I was thinking of probably generating some data sets in this kind of line. And I would probably like to get suggestions if how we can, if we were to answer this problem, what kind of ideal data sets would be for a particular disease, let's say, or in the field of, let's say, toxicity or something like that.
Yeah, so I would say, honestly, I mean, I'm less thinking about proposing new types of data, but even collecting and providing metadata for time series is not trivial. I don't know how many of you try to look for time series data. Even determining what time points were actually profiled is a huge challenge for some of these repositories. I mean, there's just no good way of querying and trying to obtain. So even the simple things, which at least to me sound simple, like what are the time points and which cells were profiled at which time point, is extremely challenging, especially if you want to do it on a large scale, like they did, as was saying in the COVID-19 presentation very nicely at the first. If you want to do this meta-analysis with time series, it would take you huge amounts of time just to get it. So I think the repository thing, even though I agree, it seems like it's easy to do, it's actually something that would require resources even before the tools and all the benchmarking, even simple questions like, you know, which gene change over time in this specific cell type. These are trivial things computationally, but extremely hard to do unless you have the information. So basically for me, treating it as a separate type of data compared to the regular or snapshot data is already a good start, which I haven't seen done in many of the repositories.
Would you mind elaborating on that? So what kind of time series data are you talking about where you can't get the metadata for the time series itself?
So just go, let's say you read a paper and it says, right, I mean, we perform time series analysis. First of all, they don't even say what are the time points, and then you go to the GSE, right? I mean, or wherever they store the data, and it's very hard to get the information sorted by whatever. I mean, the metadata capture does not always require you either to post it like that or to be able to retrieve it in a specific time series manner. If you do have time series information, you might have repeats or something or other conditions that are, so it's much harder. And, you know, we have, back in the days, we've done it more sporadically on the single cell stuff, but back in the days of RNA-seq and microarrays, we actually went and annotated the back then GEO, which was the gene expression omnibus, where everybody was required to put their, and it turned out that roughly, I think, 12% of the data sets there were time series. And I actually think there is a lot of single cell time series, even though people claim there is not. There's quite a lot, especially in the developmental, there's a lot. But it's not easy to get to them, and it's not easy, even if you do get to them, to basically understand and work with them. So again, there's no dedicated time series repository. So everything is bunched together, and I think it would be nice or useful for some people to have that, at least for me. And for developing methods, but also for querying and for other things. And I just say, HubMap actually is not interested at all in time series, so this is not something that our consortium will work on, but we'd love to see it.
So if we think that curation and benchmarking is a lot of work that is not realistic without a lot of dedicated resources, perhaps kind of two ways to cut it down into something more immediately useful and manageable could be, on the one hand, in the same way as we did for aging, maybe we should contact the metadata group to include, to make sure that they include very reasonable, very precise, very computer readable information on what is a time series data set, such that at least within the Human Cell Atlas, those, there should be a dimension for each data set. Is it time series? Yes, no, maybe. Is it a perturbation? Yes, no, maybe. And if time series, then a certain additional information would need to be annotated. So that would be kind of one way in where we would not solve the problem going backward through curation, but we would try to make sure that the meta-analysis, the metadata people are acutely aware that time series and perturbation studies are two different types of studies that are often overlaid on top of the Human Cell Atlas, and that might be very useful for certain applications outside of the context where these data were originally produced. For example, for computational method development and evaluation.
If I may, so I like this idea. I think it's a great idea. I would go a step further. So again, I'm actually in the HubMap. We're in charge of developing the computational analysis methods for the data that is being uploaded. And again, we don't have time series data, but I guess HCA has a comparable group of people that are doing that. And the thing I would stress is not just that the metadata and other annotation should be captured uniquely for time series, but also I think the treatment and the tools that are applied to it should be at least take into account time series. So we all talked about Disney and other things. That's definitely something you can do for time series, but that ignores the information on temporal differences that is already there. So I would, again, if we had a dedicated repository for time series, all the methods would have been already for time series. But if there's no dedicated repository, just the metadata, I would also argue that it would be good to have time series specific tools applied or initial processing tools, pipelines for times. At least I would love to see that. I don't know how easy it is, but I think, you know, if the metadata is there, it's possible at least to think about it slightly differently than the other. And perturbation the same way if there is any.
Then the other aspect we could do that perhaps the time series are just at this point in time not being sufficiently seen as something that is qualitatively different from the simple descriptive profiling. So obviously, kind of how it was originally written here is that we would go from data sets to benchmarking to guidelines, best practices. We could also say, okay, we think that the topic of time series and time series analysis is so important that there should be some form of review or perspectives or a paper that would emphasize the importance and ubiquity of time series, and then provide some review of existing tools and guidelines of how time series should be analyzed based on kind of the literature rather than based on a systematic benchmarking. This is a bit cutting things short, but might still have a lot of utility, both by kind of drawing attention to that time series may pop up in a lot of individual places, but are actually have some shared methodological aspect to it, and then providing very concrete suggestions of how to address those, and perhaps also motivation for then more systematic efforts that would eventually lead to benchmarking, new tool development, and really establish best practices. Is this something that people would think is valuable? And if so, is there someone who would, in principle, be interested to contribute to such an effort? That sounds like a no. Then let's move to other ideas for the time series.
Christoph, one thing you mentioned earlier was organoids, and I think maybe looking at organoids and organotypic cultures as models where you can control time and perturbations very tightly. That may be a place to begin to build some models for dealing with time series.
Yeah, I like that idea.
We talked about those opinion paper time series analysis. I mean, this idea of time series analysis, this has been around for some time. Have you thought about adding, I mean, obviously we're not the multi-omics group, right? But adding additional angles to it, potentially, you know, RNA velocity. I guess Peter may know something about this idea. Do you think this could help to chain things together, Peter, for example?
Well, sure. I mean, if you have time series, you can analyze it in this context as well. And hopefully you see some transitions between neighboring time series. You could even have algorithms that incorporate this. I think here we're mostly focusing on what's needed to try these types of things rather than the algorithms themselves, right?
Yeah, I was just thinking, like, if you want to write an opinion paper, that might be like a novel direction that, in my opinion, has been not yet maybe discussed that much.
I guess so far the focus of those people who have looked at time series, also at pseudotemporal ordering, has been for the most part in development and in immune context. So I guess the question would really be, can this be scaled beyond? Are time series a sufficiently universal concept that they can cover time series at the individual level, time series at the organ level, time series at the tissue and single cell level? And is there something to be gained from analyzing these things with similar approaches? Where I think the point has already been made that evolution is perhaps sufficiently different that it might not be yet another type of time series. But this would be like, certainly, if one wanted to pursue this idea of summarizing what is there, it seems that by going beyond the developmental aspect, one would already have some level of novelty, but also some level of additional complexity to it.
Okay.
Good, we have another 10 minutes, by which time we should really be done with it. So I see that things are already developing quite clearly on the recommendation side, and we would then still want to, like, anything else we want to add to our recommendations? And similarly, anything that we want to remove from the recommendations. And related to that, if we provide something as a recommendation, we would also have to start assigning people who are actually going to do it.
I'm also going to ask people who are interested to be listed on this consortia, please add your names and affiliations with both your institutions and specific HCA or LungMap, HubMap consortia to the section at the bottom, to the blue square, just so we will use you.
Come, I didn't get you, Peter. Alex, where do we add the name?
It's highlighted on the screen. So if you go into the doc, which is linked off the KI start page, there's this blue box, and then there's names with consortia on the team. So you could add yourself there.
So I wasn't part of any major consortium except the Chan Zuckerberg, but so would that suffice?
I guess so.
Yes, there are no limitations. Even if you are not a member of any consortia, but you would like to be, that's totally fine. Just people who are interested in continue doing this.
Okay.
Good, then in terms of kind of really next steps, I think, has anyone been involved in the metadata group in some relevant way? If kind of there's no one volunteering for that, I would try to summarize the metadata stuff, and with that contact the persons who are coordinating the metadata to try and have them take care of involving aging related topics, as well as time series, some form of time series and perturbation annotation of their data sets. Or do you have those contexts anyway? So I don't know who's in charge there. I would have to look it up and kind of email that person. Does anyone know who's in charge within the Human Cell Atlas for metadata and would be kind of in a better position than me to take care of that?
Yes, I will post this. I will add this to the document. I have a contact person who runs this from Sanger. They have stable metadata meetings almost every other week.
Okay, great. So you would bring in the points that we've written down here, including annotation of any aging-related information, the possibility of adding epigenetic clock information on peripheral blood, and also the denoting studies as time series and or perturbation as much as possible into the meta standards. Great. Any other points that we should take forward and people who would actually do that? Do we want to, we had several times came up the topic of, for certain things, we would need certain kind of, like we would pitch new ideas, certain ideas that people would, that would be useful to do if there's funding available. So obviously we have summarized it here to some degree. Would there be value in extending that further and providing kind of more like a 2-paragraph summary or a couple of bullet point summary that goes beyond that? Or do you feel what we have here is good enough and we can rely on the people seeing it in these documents? Good, I guess that's a point that applies mainly to the NIH or US-based folks. So, and they are looking at these documents anyway, so we're probably fine if we leave it like that.
I have something I just thought about. I know that most of these methods, they really depend on the single cell data sets being well pre-processed and everything. So I don't know how, if somebody has studied the impact of this upstream and processing to dynamic, because at the end of the day, these methods or the analysis of the dynamic aspects of the data really depend on the pre-processing of these data sets. And I don't know if that is an important thing to consider before really applying some of the or modeling the data.
Okay, then it seems that by and large, we have a summary. It also seems that we have, like how I read it at the moment, we don't, I guess the key step that we need to do going forward is we need to really pitch towards the metadata group that the importance of aging, the importance of time series, and the importance of perturbation annotation and the importance of epigenetic clocks. And with that, we should already be quite far. And otherwise, it doesn't seem like we have something that we would necessarily have to pitch within the plenary session as the outcome of this, or are there any points that you would think fulfill the key points of, let me check, what was that? It should be something new about the collaboration, new challenge, new approach, new parties, and you have passion and plan to meet again, and you have to ask something from the community. Would we have anything here that should get a 2-minute plenary response, or is it sufficient that we have it written up here in our results section?